In our last screencast on Chapter 9, we're going to talk about anaerobic respiration. Okay, so what happens when there is no oxygen present during the process of, after the process of glycolysis. And this is also referred to as fermentation. So most cellular respiration that we've talked about thus far requires oxygen in order to produce ATP. Oxygen needs to be present for pyruvate to be transported into the mitochondria at the end of glycolysis and yield a large amount of ATP through the electron transport chain. However, when there's no oxygen, the electron transport chain doesn't work. So in the absence of oxygen, fermentation and anaerobic respiration allow cells to continue to produce ATP via glycolysis. So when there's no oxygen, glycolysis is our only source of ATP. All right, so anaerobic respiration uses a terminal electron acceptor other than oxygen. It doesn't use oxygen, it uses something else, for example, sulfate or something like that. Fermentation uses substrate level phosphorylation instead of electron transport chain to generate ATP, which is why it is so little ATP. Very little ATP produced. Because it only produces those two that we get in glycolysis. So fermentation is glycolysis plus a reaction that regenerates NAD+. And then the NAD+ can be reused by glycolysis. So basically, the main purpose of fermentation, P-U-R-P-O-S-E, of fermentation is to regenerate NAD+. Okay? That plus is a little messy. NAD+. Okay. So there are two types of fermentation, one alcohol fermentation and two lactic acid fermentation. So first we're going to go through alcohol fermentation, and this happens in yeast or other types of organisms, but not animals. Okay, so alcohol fermentation is then exploited because it produces some alcohol. This is brewing, winemaking, baking, making bread, and you'll see why in just a minute. Okay, so in alcohol fermentation, pyruvate from glycolysis is converted to ethyl alcohol in two steps, and the first one releases carbon dioxide. So let's have a look. So here we have glucose going through glycolysis and yielding pyruvate. Okay. Over the course of that process, we get a net of 2 ATP produced, and we also consume 2 NADH and produce 2 NADH2. So if there's no oxygen, we have to regenerate that NAD+ so that glycolysis can continue. So if there's no oxygen, pyruvate is not transported into the mitochondria, but it goes through this process of alcohol fermentation. Move this up a little bit. Where it gets converted into ethyl alcohol in two steps. The first step releases some carbon dioxide, okay, to form acetaldehyde, and then that gets converted, consuming the energy in NADH into ethyl alcohol. Okay, that then regenerates my NAD+, and glycolysis can use that to continue to make our ATP. Okay. Now this process, if you think about baking or making beer or wine, right? So the carbon dioxide release, that's a gas, right? It's bubbles, and it makes that's what makes dough rise, as all the bubbles of carbon dioxide released by fermentation of the yeast, and that's what makes all those little holes in your bread. Those are made by bubbles of carbon dioxide. When you bake bread, you bake off all of the ethyl alcohol, which is why you can't have alcohol by eating bread, because it gets baked off. However, when you're making beer or wine, right, so the bubbles in the beer also come from the carbon dioxide as well as the alcohol content. Okay, so that's alcohol fermentation. Our second type of fermentation is lactic acid fermentation. And you may have heard your coaches talking about lactic acid buildup in your muscle. That's because this is the type of fermentation that animals do when we don't have enough oxygen in order to keep that ATP being made. So lactic acid fermentation, pyruvate is reduced, and lactate is the end product. Okay, but there is no release of carbon dioxide. Okay, lactic acid fermentation by fungi and bacteria is used to make cheese and yogurt. That's why it can have that zippy taste. And human muscle cells also use lactic acid fermentation to generate ATP when oxygen is low. Okay, so here is a look at lactic acid fermentation. Nudge that up a little bit more. Oops. Okay, so again, here we have glucose going through glycolysis to make pyruvate. Okay, this generates our ATP and NADH. Now, again, there's no oxygen. Pyruvate does not go into the mitochondria, and it gets converted in the cytosol back to, or into lactate. That process converts NADH back to NADH2, which can then be used in glycolysis. Okay, and again, that's all this is occurring in the cytosol. Okay, as well, alcohol fermentation also occurs in the cytosol because nothing, when there's no oxygen, nothing gets transported into the mitochondria. Okay, so let's compare these two processes. So aerobic respiration is, of course, way more efficient. It gets way more of the energy out of glucose. So both processes use glycolysis, and that makes me 2 ATP, to oxidize glucose. Okay. In all three processes, so fermentation, anaerobic, and aerobic respiration, NAD+ is required as an oxidizing agent that accepts electrons during glycolysis. Okay, the processes have different final electron acceptors. Okay, so in fermentation, it's an organic molecule, and in cellular respiration, it's oxygen. Okay, cellular respiration produces a whole bunch of ATP per glucose molecule, and fermentation produces much lower number, but still, it is a net gain of ATP, and so we have to still produce ATP, and 2 is better than 0. Okay, a couple of different types of organisms that undergo anaerobic respiration. There are things called obligate anaerobes, and think about if you're obligated to do something, you have to do it. So obligate anaerobes only use fermentation or anaerobic respiration, and they cannot survive in the presence of oxygen. So they have to do anaerobic respiration. Oxygen will kill them. Yeast and a lot of bacteria are facultative anaerobes, means they can survive using fermentation or cellular respiration. Okay, and facultative anaerobe, pyruvate is kind of this decision point in a metabolic road, a fork in a metabolic road that leads to 2 alternative catabolic routes. So if you look here, if I have some glucose, glucose always undergoes glycolysis. Every single organism living on the face of the planet uses glycolysis. Okay? At the end of glycolysis, we have our pyruvate. And then at this point, the cell makes a decision. If there is no oxygen present, it goes to anaerobic respiration, so ethanol, lactate, or other products. Okay? If there is oxygen present, we go through aerobic cellular respiration because it's preferred, because it makes more ATP. Okay? Just a couple of, bring in our evolution piece, evolutionary significance of glycolysis. Glycolysis is an extremely ancient process. It's one of the first metabolic processes they believe evolved, period. Okay? Ancient prokaryotes were thought to have used glycolysis long before there was oxygen in the atmosphere. Remember, glycolysis doesn't require oxygen. So it got the cells energy before there was even oxygen in the atmosphere. And that was a really long time ago, like 3 billion years ago. Oxygen became available about 2.7 billion years ago. So early prokaryotes likely only used glycolysis to generate ATP. Okay, and that's it for glycolysis, for fermentation, for aerobic respiration, and for chapter 9.